Okay, we're going to pick up our next podcast here, continuing with the looking at the evidences for evolution. We talked about fossils and evaluating the fossil record last time. In this lecture, we're going to look at about four different types of other types of evidence that's used to support evolution. And again, we need to distinguish that we are talking about macroevolution, that historical evolution. There is no need for evidence for microevolution. It's observable. We can see that occurring. So again, we're talking about in the past the macroevolution. The next line of evidence we're going to look at is homology. Homology is the study of similar structures. Homo means the same, um, and so this is the study of similarities of structures. And this is often used to support a view that these similar structures relate back to a common ancestor. So this is a very common figure that's used in this line of evidence. This is a forearm, human forearm, front leg of a cat, I believe, front flipper of a whale, and the wing of a bat. And if you look at these structures, there's a lot of similar design here, a lot of similarity in these structures. Two forearm bones here, two bones there, two bones there, two bones there. And if you look, there's very similar pattern here as you look at those four different animals. And so the logic goes that these similarities um, go back to supporting a view that they all had a common ancestor somewhere in the past. So this is certainly one perspective of the structures being homologous, that they have a common ancestor in the past. There is other perspectives, such as that of the intelligent design proponents that we spoke about earlier. And that would be, those proponents would say these common structures or similar structures could be related to a common design plan. So that would be two explanations for these similarities in these structures, would be either common ancestry or common design. Um, this could also be applied to um, embryos, and looking at and comparing embryos of different animals, the similarities that we see in frogs and birds and cats and humans in that embryonic development, those early stages. Certainly there would be similarities. Every living thing starts out as one single zygotic cell, and so those cells, of course, are going to be very similar, and then through that embryonic development, you have some similarities that would be occurring in similar looking animals. So applying that would be a form of comparing those homologous structures. And in applying this to embryos, it's sometimes even taken further. Ernest um, Haeckel's embryos, biology textbooks often represent as comparing these embryos of different animals, but Haeckel um, developed what he called the biogenetic law, and that is that ontogeny recapitulates phylogeny. So that phrase, ontogeny recapitulates phylogeny, um, is representing the ontogeny, which is the development of the embryo, is a copy of phylogeny, which is a evolutionary development of the organism. So what he is saying is that as you track the embryonic development, you're seeing kind of a snapshot or a picture of the embryonic or the evolutionary development of that individual. So if humans have these pharyngeal pouches in their embryonic development, and these pouches have some similar resemblance to gills, and so he would say that that is showing a picture of gills being in the evolutionary past of humans. And so that's kind of comparing that in the embryos to the evolutionary development of the individual. Also closely aligned to this idea of comparing structures would be in the idea of vestigial structures, and these are structures that are said to have lost a function from the past. Say, for instance, the appendix or things like this, the pineal gland, the thyroid gland, the tonsils. You can have these taken out and still function properly. So the theory is that somewhere in the past these may have had a stronger function, and so maybe they're a leftover remnant from an evolutionary ancestor. Um, so those are all things in kind of comparing structures in that exist today in animals and trying to make some inference or ideas about their past link to evolutionary relatedness or common ancestry. Um, all right, so another line of evidence that we can look at is very similar to homology, but it's not comparing structures, but rather comparing uh, not physical structures, but comparing molecules within the organism, or comparing the structure of the molecule, DNA or proteins that the organism carries. And so here's one your book shows right here. And this one is comparing the hemoglobin protein here of these different animals right here. And so what they're comparing is the sequence of the amino acids. So humans as the starting point, these monkeys would have whatever number that is, percentage similar in the amino acid sequence, and then mice right here and so forth. As you go down, the amino acid sequences become less and less similar. And so the logic behind this is this exact same logic that is used in homologous structures, and that is that this similarity, because these have a closer percentage of these amino acids in that particular protein, that similarity would imply a common ancestor, that they're more closely aligned with each other. And this works good for a line of evidence of evolution in some cases and not good in other scenarios. There are some proteins that relaxin is an example of a protein that we see in a lot of different mammals and other animals as well. And this protein has more similar types of amino acids in the in the pig and the shark rather than the pig and the rodent. And pigs and rodents should have more similarities to them because they are in the same group. So the similarity of structures is not always showing you the most close relatedness, but that's how it's usually used, as similar structures relate to similar ancestry. So your book mentions another example of evidence that's used for evolution, and that is biogeography. So biogeography defined as the distribution of species, so the geographical distribution of species on the continents of the Earth. So the theory is primarily that the Earth was at one time just had one giant landmass, and then this landmass, um, which is has been given a name Pangea. So this theoretical landmass split into smaller landmasses, ultimately into the continents that they are today. And animals that were on those continents, became isolated and um, no longer were in contact with the other populations. And so over time, you see specific traits becoming isolated and becoming more dominant at different continents. That's why mammals, say in Australia, are different. You have more marsupials and things like that there, whereas you have placentals here in America. And so the theory is that those changes or those differences can be explained by their geographical distribution of the pattern. Um, Now, biogeography in itself is not an explanation for where these traits came from. It's just the idea that the isolation, um, uh, gave certain individuals better opportunity to survive. Um, and so it's more of an explanation of why we see more animals in some continents and not some in other continents. Now, Darwin recognized this and promoted this as he spent his travels on the when he took his travels around on the HMS Beagle. 3 years he spent on that, traveling around the globe, recognizing these different types of plants and animals on different continents, based on the climatic condition and the food availability and things like that. He would see these very different animals on the different continents, and so he developed a lot of his idea based on biogeography. Um, so that is the lines of evidence that are used to support evolution, macro or historical evolution. The next podcast we'll be going into is going to be looking at the view of the mechanism of evolution, and that is natural selection. Darwin wrote a lot about this, and so we'll be looking at that in the next podcast.